The virus responsible for COVID-19 invades human cells thanks to spike proteins on its surface that bind with high affinity to the ACE2 receptor on the human cells. Once bound, the virus is engulfed and enters. Now protected from the immune system inside the cell, it is free to replicate and release fully functional SARS-CoV-2 cells that then exponentially repeat the cycle. It follows then that disrupting this entry would neutralize the virus by leaving it accessible and vulnerable to the human immune system outside the cell. SignalChem has successfully manufactured the proteins needed for the strategies presented here that would do just that, and are asking for collaborators to use them in the pursuit of clinical trials, treatment, prevention, and IVD development. One strategy involves the spike protein, which on its own cannot cause infection. Free spike protein would occupy the ACE2 receptor, preventing the virus from binding. In addition, it would also trigger an immune response that would prepare the body for future infections, similar to a vaccine. But blocking a functional channel could result in unwanted side effects. A similar approach would involve antibodies to the ACE2 receptor that would physically hinder the virus from binding. But again, the functional channel would also be affected. In collaboration with Austrian in silico powerhouse Innofor, variations of the extracellular domain of ACE2 were designed and manufactured that have a higher affinity to the spike protein. The presence of these proteins could effectively encapsulate the virus and prevent invasion. And as another example of the potential in collaboration, a Canadian consortium of Novobind, Cedar Lane, Natural Products Canada, and SignalChem, with legal help from US-based Wilson Sonsini,